welcome to the Deep Dive. Today we're tackling a topic that's, well, fundamental to the safety of pretty much every pill you might take. Cleaning validation in pharmaceutical manufacturing.
It really is. Think about it. Multiple different medications often made on the exact same equipment.
Exactly. So we're going to explore the incredibly meticulous steps, the thinking that goes into making sure that equipment is thoroughly, provably cleaned between batches.
It's absolutely critical. When a cleaning process fails, even slightly, the consequences for patient safety, they can be huge.
You mean like trace amounts of one drug ending up in another?
Precisely. Especially for someone with allergies or maybe taking specific drugs where interactions could be really harmful. That's the high-stakes reality cleaning validation is designed to prevent.
Right. And that's really the mission. What exactly is this cleaning validation master plan? Why is it such a cornerstone document?
Well, think of it as the rule book. It's the central document that spells out exactly how a pharmaceutical company guarantees its cleaning procedures actually work. Every single time.
It's not just a suggestion, then.
Oh, definitely not. It's a formal, living document. It needs official sign-off, signatures and dates from various departments. It shows everyone's on board and accountable.
Okay.
And the core purpose, like this plan lays out, is to provide absolute assurance that those cleaning procedures completely remove any residue.
Residue from the previous drug.
Yes, from the previous product and also from any cleaning agents used. And they have to remove it down to levels so low they pose absolutely no risk to patients taking the next product made on that equipment.
So it's basically creating this invisible shield, making sure nothing unwanted carries over. Now, this particular rulebook, this plan we're looking at, it focuses on a specific area.
It does. This master plan is specifically focused on the tablet manufacturing department at Pharma.
Ah, okay.
Tablets. And that's an important distinction because the cleaning challenges, the considerations, they can differ quite a bit depending on the type of product. Cleaning an ointment line is very different from cleaning tablet presses.
Right, different forms. Stickier residues, perhaps. Now, the plan uses a couple of key terms right up front. Worst case product and cleaning validation itself. Can we break those down? What makes a product the worst case?
Okay, the worst case product. It's a deliberately chosen representative from all the different drugs made using a same set of equipment.
How do they choose it? Just pick the hardest one.
It's more scientific than that. It's based on a careful evaluation of several factors. First, there's a toxicological assessment. How potentially harmful is the active ingredient?
Okay, toxicity.
Then, solubility in water is key. How easily does it dissolve? Because, that affects how easy it is to wash away.
Makes sense.
Another crucial factor is the lowest therapeutic dose. If a drug is really potent, even tiny remnants could matter.
Right. A little goes a long way.
Exactly. And finally, they consider its general cleanability, things like how sticky it is, or if it's colored, or just known to be difficult to remove.
So it's kind of like identifying the absolute toughest stain to make sure your cleaning method can handle anything. If it works for that, it should work for the easier ones.
That's a great analogy, yes. If you can prove your cleaning works after making the worst case, you've got high confidence it works for the less challenging ones.
Okay, and cleaning validation itself, the term.
Cleaning validation, as defined here, is the whole system. It's the documented evidence that provides that high degree of assurance we talked about. It's the methodology used to show, reliably, that the cleaning process removes not just the active ingredients, but also any cleaning agents, and controls microbes.
Okay, that definitely paints a clearer picture of why this plan and the whole validation process are so critical. Now, who actually does all this? Who are the different teams involved? It sounds like it needs a lot of different types of expertise.
It absolutely does. This master plan outlines several key departments and their specific roles. Not surprisingly, quality assurance, or QA, is right at the center.
QA, right.
They're responsible for preparing and approving the master plan itself, and also the individual validation protocols, the final reports. They also plan and monitor all the validation activities, kind of the oversight role.
So QA is keeping an eye on everything, making sure it's done right and documented properly. What about the teams actually doing the cleaning and working with the equipment?
That's where production comes in. They develop and approve the specific step-by-step cleaning procedures for each piece of equipment.
They know the equipment best, presumably.
Exactly. And they bring that practical knowledge, helping to identify, say, tricky spots that might be harder to clean, and of course they arrange for the cleaning to actually happen.
Okay. Any other key players?
Oh, yes. Engineering is vital. They also help identify those tricky-to-target locations from a design standpoint, and crucially, they calculate the product contact surface area of all the equipment.
Surface area. Why is that so important?
Because that number, the total area that touches the product, is absolutely essential for setting the acceptable residue limits later on. You need to know the total area you're trying to clean.
Okay. To figure out how much residue is acceptable per square centimeter, for example.
Precisely. And then there's the testing part that falls to quality control, or QC, both the chemical lab and the microbiology lab.
The lab's doing the analysis.
Right. They're the analytical powerhouses. They develop and validate the highly sensitive methods needed to test the samples taken during validation. They do the actual testing and generate the reports showing whether the cleaning met those stringent targets.
Okay. QA, production, engineering, QC, anyone else?
We also have research and development, R&D, and technology transfer involved. They bring deep knowledge about the products themselves.
How they behave.
Yes, how easy or difficult a specific product might be to clean, any special cleaning needs based on its chemical or physical properties. They share that crucial information.
It really is a team effort. And there's one more group mentioned, the toxicologist. What's their role in cleaning? Seems a bit unusual.
It might seem so, but the toxicologist is absolutely essential. They provide the critical toxicological evaluations for all the active pharmaceutical ingredients, the APIs.
Back to the worst case selection.
Yes, that information is key for identifying the worst case product. But maybe even more importantly, they calculate something called the permitted daily exposure, or PDE value, for each API.
PD, permitted daily exposure.
Right. That PDE value is fundamental. It's used to scientifically set the limits for how much residue can safely be allowed to remain after cleaning.
So they determine the actual safety threshold based on potential harm.
Exactly. And they also keep up with the latest science, routinely reviewing toxicological data to make sure those safety limits are always based on the most current understanding.
Wow. It really is a cross-functional effort then. Everyone from the shop floor to the lab to the toxicologist is involved in ensuring this cleaning is effective and validated.
Absolutely. Every angle is covered.
Okay, let's shift gears a bit. What about the guiding principles? What are the core ideas, the tenets, that underpin this whole cleaning validation policy?
The policy really sets the stage. It outlines the essential steps needed to get consistently effective cleaning. Things like the careful selection of that worst-case product and the relevant equipment.
We've covered that.
Right. Then determining the best sampling methods and where to take those samples, choosing analytical techniques sensitive enough to find trace residues.
The QC part.
Yes. And conducting those residue recovery studies, we need to make sure our sampling actually works, and then finally doing the actual sampling during the validation runs.
Okay, so those are the key stages. What's the big picture goal here? What's the policy ultimately trying to achieve?
Well, the number one goal, as we've sort of touched on, is the absolute prevention of contamination and cross-contamination, especially critical in a multi-product facility.
Makes sense.
The policy emphasizes monitoring for 3 types of contamination: chemical, which is the API residue,
Main drug.
physical and any visible particles or impurities, and microbial contamination from bacteria, mold, things like that.
So they're looking beyond just the chemical traces.
Definitely. And to monitor these, they use a combination of methods. First, that crucial visual inspection. If you can see something, it's not clean.
The first check.
Then, sophisticated chemical testing to detect tiny amounts you can't see, and specialized microbiological techniques to ensure no harmful microbial growth on the surfaces.
Okay.
And a core principle here is that all product contacting critical processing equipment must have validated cleaning procedures. No exceptions for equipment that touches the drug.
If it touches the product, it needs proofs the cleaning works.
Got it.
The policy also mentions risk assessment. How does that fit in?
Risk assessment, often using quality risk management or QRM principles, is woven throughout. It's a systematic way to evaluate the potential for contamination at each step and to check how effective our controls are.
Controls like?
They can be technical, like designing equipment to be easy to clean, or the specific cleaning procedures themselves, or they can be organizational, like planning production campaigns to minimize changeovers, or the verification steps after cleaning.
So QRM helps focus efforts on the biggest risks.
Exactly. It's a proactive approach. Identify potential weaknesses and address them before they cause a problem, especially important in such a high-stakes area.
Makes perfect sense. So we've covered the planning, the policy. What does the actual cleaning process typically involve? And what needs to be sorted before you can even start validating it?
Right, before any cleaning validation can even begin, several foundations have to be solid. The equipment itself must be qualified, basically, proven to work consistently as intended.
Checked and certified.
Yes. The utilities used in cleaning, especially the purified water, must be verified to meet strict quality standards. Impure water won't clean properly.
Good point.
And any critical instruments used either in the cleaning process or for testing afterward must be calibrated. Accurate measurements are essential.
So, like ensuring your tools are sharp and accurate before starting a complex job.
Exactly. Get the basics right first.
Okay, groundwork solid. How are the cleaning procedures themselves developed?
It's usually a mix of inputs. Information might come from the site transferring the product or from R&D. Equipment manufacturers often provide suggestions too. They know their machines best.
Manufacturer recommendations, okay.
And then there's the company's own past experience. Lessons learned from cleaning similar equipment or products before. It's a blend of science, supplier knowledge, and practical experience.
Right. Now, this plan is for tablet manufacturing. Can you give us a quick rundown of that process just so we can picture where cleaning fits in?
Sure. Tablet manufacturing, it's usually multi-stage, starts with sifting raw materials for the right particle size, then mixing, often in a granulator, sometimes adding liquids to form granules. That's wet granulation.
Granulation.
If you wet granulate, you then need to dry the granules, often in a fluid bed dryer, then maybe size them again. Sometimes lubricants are added to help with the next step.
Which is?
Compression, using a tablet press machine to form the actual tablets. After that, depending on the tablet, it might get coated. Then finally, packaging.
Lots of steps, lots of different equipment touching the product.
Exactly. And cleaning is critical after every batch of one product before you switch to a different product on the same equipment.
Okay, so what does that cleaning process generally look like according to this plan?
The plan outlines a general flow. It often starts with dismantling the equipment as much as practical to get at all the contact surfaces.
Taking it apart.
Then usually an initial rinse with potable water, drinking water quality to remove the loose stuff, followed by a more thorough scrub, often still with potable water, maybe using brushes.
Getting into the nooks and crannies.
Right. Then another potable water rinse. The final step is typically a rinse with purified water that removes any residues from the potable water itself.
Ah, an extra level of purity.
Yes. And then, crucially, the equipment is thoroughly dried, often using filtered compressed air or a dedicated dryer.
Drying seems important.
Very. Residual moisture is a breeding ground for microbes, so getting it completely dry is key for controlling that third type of contamination microbial.
So the rinsing and scrubbing handles the chemical and physical stuff, and drying tackles the microbial risk.
Broadly, yes. The water steps physically remove residues, and the purified water rinse ensures high chemical purity. Drying inhibits microbial growth. For really tough residues, they might use specific validated cleaning agents or more intense scrubbing.
And the validation proves this whole process actually works for all three contamination types.
Precisely. The validation confirms effectiveness through that visual check, the chemical testing, and the microbiological sampling we talked about.
Got it. Now, going back to that worst-case product idea, how do they actually identify it systematically? It sounds like quite an analytical process.
It really is. The master plan describes a structured approach, sometimes called product grouping, or creating a product matrix.
A matrix.
Yeah, basically listing all the different products manufactured and grouping them based on which specific pieces of equipment they share during production.
So you map out which products potentially cross paths on the same machinery.
Exactly. This includes commercial products, but also maybe products still in development or used for registration batches, though usually commercial and pilot development areas are grouped separately.
Okay, you've got your group of products sharing equipment. Then how do you pinpoint the worst case within that group?
It's a multi-factor assessment. First is that toxicological assessment we mentioned. APIs are often categorized into hazard groups like relatively safe, low hazard, moderate hazard, highly hazardous.
Based on potential harm.
Right. For the relatively safe and low hazard ones, the focus then shifts more to those product characteristics that make them hard to clean.
Like solubility or stickiness.
Exactly. Moderate hazard products need stronger controls anyway, and highly hazardous ones, well, they might even need dedicated equipment or separate facilities altogether to minimize risk.
Makes sense. You wouldn't want, say, a potent chemotherapy drug run on the same line as a simple vitamin without extreme measures. What makes something highly hazardous?
The plan lists specific criteria. Things like known or suspected carcinogens, substances causing reproductive harm at low doses, those causing serious organ damage at low doses, or those with a high potential to cause allergic reactions.
Pretty serious stuff.
Yes. And it also mentions that highly potent compounds, those effective at very low doses, are assessed carefully too, often based on their therapeutic dose rating, because even tiny amounts could have a pharmacological effect.
So potency itself is a red flag. Now, you mentioned PDE earlier, permitted daily exposure. That seems central here. Can you unpack that a bit more? How is it determined?
Okay, PDE, permitted daily exposure. It's a health-based limit. Essentially, the maximum daily dose of a substance someone could be exposed to over a lifetime without expecting adverse health effects. It's calculated for every API.
Oh.
It starts with toxicological data, usually the NOAEL, the no observed adverse effect level. That's the highest dose tested in studies, often animal studies, where no harm was seen.
The highest safe dose found experimentally.
Kind of. If a clear NOAEL isn't available, they might use a no observed effect level, or even LOEL, lowest observed effect level, but they'd apply bigger safety factors then.
Safety factors.
Yes. The formula takes the NOAEL, adjusts for body weight, and then divides by several uncertainty factors, often called F1 to F5.
Well, what do those account for?
Things like differences between animals and humans, variability between individual people, how long the study lasted, how severe the toxicity was, whether it was a true no effect level. It builds in safety margins.
Lots of layers of caution.
Absolutely. And crucially, the plan says they have to keep reviewing scientific literature. If new data comes out that changes the understanding of the NOAEL or toxicity, the PDE has to be reevaluated.
So it's not static, it adapts to new science.
Correct. Which could potentially trigger a reassessment of the cleaning validation itself.
Okay, that's a rigorous way to set safety limits. Now, for those low-risk products, how do they pick the worst case based on cleanability?
For those, they use a rating system based on 3 key attributes. First, solubility in water. Less soluble gets a higher, worse rating.
Harder to wash off.
Second, the lowest therapeutic dose. Lower dose gets a higher rating because smaller amounts matter more.
More potent.
And third, a general cleanability rating. This considers practical things. Is it colored? Is it sticky? Does R&D or past experience suggest it's tough to clean?
So solubility rating A, potency rating B, and general difficulty rating C.
Exactly. They multiply those ratings. A times V times C gives a total risk value for each product in the group.
And the highest score wins. Or loses, rather, becomes the worst case.
Precisely. The product with the highest total risk value is selected, if there's a tie.
Tiebreaker rules.
Yes. First tiebreaker is the most insoluble product. If still tied, it's the most potent, lowest therapeutic dose. And this whole matrix gets reviewed whenever new products or equipment come in.
Very systematic. What about the equipment itself? Is there a similar worst-case idea for equipment?
Yes, similar concept. They create an equipment matrix, grouping similar types of equipment together, machines with comparable design, size, complexity.
Grouping similar machines.
The idea is if you validate cleaning on a representative piece from that group, you can be confident it works for the others in the group.
Yeah.
Saves validating every single identical mixer, for example.
Makes sense. But what about dedicated equipment used for only one product?
Ah, good point. Dedicated equipment or parts are usually excluded from this cross-contamination-focused cleaning validation because they don't pose that risk. They still need cleaning and microbial control, just not the same multi-product validation burden.
Okay, so identify worst-case product, maybe representative equipment.
Okay.
Then how do you know if the cleaning actually worked well enough? What are the targets, the acceptance criteria?
This is absolutely key, the acceptance criteria. For chemical residue from the API, there are several criteria, and the strictest one always applies.
Strictest limit wins, okay.
First, hurdle, visual clean. After cleaning and drying, a thorough visual check, maybe with a bright light, must show 0 visible residue. If you see anything, it fails right there.
Can't look dirty, basic standard.
Exactly, prerequisite. If it passes visual, then they move to quantitative chemical testing against MACO limits. Maximum allowable carryover. How much residue is allowed to potentially carry over? It's calculated in a few ways.
Okay, what are the ways?
One is based on the daily dose. The idea is that no more than 0.1%, 1,000th of the previous product's normal dose should appear in the maximum daily dose of the next product.
A tiny fraction of a dose.
A very
tiny fraction of a dose.
A very tiny fraction. There's a formula using therapeutic dose, batch sizes, surface area, dosage units to calculate this limit, often in milligrams per 100 square centimeters for a swab sample.
Okay. Dose-based limit. What else?
Another is the 10 ppm criterion. Simple principle. No more than 10 parts per million of any product residue should be found in the next product. Again, a formula converts this to an amount per swab, considering batch size and unit dose.
10 parts per million, a general concentration limit.
Right. And then, crucially, they also calculate MACO based on that PDE value from the toxicologist.
Ah, the health-based limit.
Exactly. This uses the PDE to determine the maximum residue amount consistent with patient safety. The formula considers the PDE, standard body weight, batch size, daily dose of the next product, and surface area.
So dose-based, concentration-based, and health-based calculations.
Correct. And the plan is clear. You calculate MACO all these ways, and you must use the lowest, most stringent value as your acceptance limit for the validation.
Highest level of safety. Makes sense. Is that just for swabs? What about rinse samples?
They have a formula to calculate the equivalent MACO limit for rinse samples, too, based on the swab limit and the total equipment surface area. That's used when you can't swab effectively.
Okay. What about the other contamination types? Microbial.
Yes, there's a limit for that, too. For microbiological examination using sterile swabs, the criterion here is not more than 30 colony-forming units, CFU, per 100 square centimeters.
So controlling bacterial or fungal load, too.
Absolutely. And they even set limits for any residual solvents used during sampling, often using a similar PDE-based calculation for rinse samples to ensure the sampling itself doesn't leave harmful residues.
Wow. They really cover all the bases. Drug residue, microbes, even the testing chemicals. So how do they actually get these samples? Swabbing and rinsing.
Exactly. Those are the two main methods detailed: swab sampling and rinse sampling.
Swabbing seems pretty direct.
It is. You use a swab moistened with a specific desorption solvent that dissolves the target API and wipe a precise, defined area, often using a 10 by 10 centimeter template.
Why use swabs?
They're great for getting a specific contamination status from accessible surfaces, especially hard-to-clean spots or areas where residue might gray on. The swab then goes into a vial with more solvent to extract the residue for the lab.
Targeted wipe test. What about rinsing?
Rinse sampling is used for surfaces you can't easily swab inside pipes, complex internal parts, equipment not easily dismantled, or just very large surface areas.
How does that work?
You rinse the surface with a known volume of a suitable solvent one that dissolves the API but is also water-soluble itself, so you can wash it away later. Collect a sample of that rinse water for analysis.
And you mentioned rinsing for solvent traces, too.
Yes, rinse sampling is the main way they check for leftover desorption solvent residues. And for microbial testing, it's always swab sampling with sterile swabs.
Okay, swabs for direct, accessible spots and microbes. Rinses for inaccessible or large areas. How do they choose where exactly to sample? Can't be everywhere.
No, it's strategic. For chemical residues, they focus on known hard-to-clean areas.
Like what?
Corners, joints, welds, pipe bends, complex shapes, places where product might pool or flow slows down, parts hard to take apart. Also, spots that might be physically awkward to reach during cleaning.
The usual suspects for where dirt hides.
Pretty much. They aim for enough locations to give high confidence that if residue is hiding anywhere above the limit, they'll find it. For small, simple equipment, they might sample the whole surface. If swabbing is impossible in a critical spot, a rinse sample might substitute.
Makes sense. What about choosing microbial sampling spots? Same logic.
Similar risk-based thinking, but focused on where microbes might thrive. Areas prone to residual moisture, crevices, rough surfaces, places likely to get contaminated during use, spots known to support growth, or difficult to disinfect areas.
Different factors for microbes versus chemicals. Is there a specific order for taking these different samples?
Yes, usually. To avoid cross-contamination during sampling, a typical sequence might be rinse sample for cleaning agent first, then microbiology swabs, then API swabs, maybe then an API rinse sample.
So you don't want the API solvent messing up the microbe sample, for instance.
Exactly. Sequence matters.
What about the swabs and solvents themselves? Any special requirements for those materials?
Oh, yes. The swab material can't interfere with the lab test, needs to be compatible with the solvents and the API, low linting so it doesn't shed fibers, and must allow good recovery of the residue.
And the solvent.
The desorption solvents need to dissolve the target API well, be compatible with the equipment surfaces and the swab, and not mess up the analysis. If using organic solvents, they have to check with the equipment supplier. It won't damage anything.
And make sure the solvent itself is gone afterwards.
Absolutely. There must be procedures to ensure residual solvents are removed before the next product run. The equipment is usually cleaned again after sampling to remove any traces of sampling solvents.
Thorough. Now, we touched on product-dedicated parts equipment earlier. Why are they treated differently again?
Right. Some parts, or whole machines, might be just too difficult to clean reliably for multi-product use, or too hard to accurately test for residues.
So they just dedicate them.
Yes, designating them for only one specific product eliminates the risk of cross-contamination between different products. So they're excluded from the cleaning validation program focused on that risk.
But still cleaned.
Oh yes, they're still part of general hygiene and microbial monitoring programs, just not the multi-product validation protocol. It's a risk management strategy.
Got it. Now, validation isn't just about one perfect clean. They simulate real-world conditions, too, like delays.
They do. One key simulation is the dirty equipment hold time.
Dirty hold time.
Yeah, intentionally leaving the equipment dirty, uncleaned, for a set period after making the worst-case product before starting the cleaning procedure.
Why do that?
To mimic real-world delays. Maybe cleaning can't start immediately due to scheduling. This challenges the process. Does the residue become harder to remove if it sits there? They usually do this for at least one of the 3 validation runs.
Stress testing the cleaning against delays. What about time after cleaning, before reuse?
That's the clean equipment hold time, the maximum time allowed between finishing the clean and starting the next batch.
Why limit that?
Primarily for microbial control, to prevent significant microbial growth on the clean stored equipment. This time limit is established and supported by microbial monitoring data.
So checking robustness at both ends, how long it can sit dirty and how long it can sit clean. Now, measuring these tiny residues requires super precise lab methods, right?
Absolutely critical. After selecting the worst-case product, the analytical methods for detecting the API, any cleaning agents, any sampling solvents, they all have to be developed and rigorously validated.
Validated how? What do they need to prove?
The method validation proves the test is reliable, accurate, and fit for purpose. They test parameters like specificity, does it only detect the target? Limit of detection and quantification, how low can it reliably measure?
Sensitivity.
Linearity, does the signal match the amount? Accuracy, how close is the result to the true value? Precision, how repeatable is it? Range over, what concentrations does it work? Stability, do the samples degrade over time?
So, proving the test method itself is scientifically sound and sensitive enough.
Exactly. It must be able to detect the target substance at or below that strict acceptance limit we set. It needs to work even with other stuff present in the sample. And they need to confirm the API is stable during analysis.
And you mentioned recovery studies earlier, too. That's part of method validation.
It's closely linked. The surface recovery studies, or spike recovery, demonstrate how well the sampling method, the swab or rinse, actually pulls the residue off the equipment surface before it even gets to the validated analytical method. And the extraction method from the swab rinse also needs validation.
Lots of steps to ensure the final number is trustworthy. How do they choose which analytical technique to use? HPLC or something else?
Practicality plays a role. They often prefer simpler methods, using equipment already available and validated at the site. Sample preparation should ideally be easy and quick, but the method must meet all those validation criteria: sensitivity, specificity, accuracy, precision, and the instruments must be calibrated.
Okay, practical but scientifically robust. Let's dive into those residue recovery studies again. They sound crucial for knowing if the sampling itself works.
They really are. Purpose is to check how well the sampling method swab or rinse actually recovers the residue from the specific equipment surfaces, because you won't get 100% recovery in reality.
How do they do it?
They take clean coupons, small pieces, of the actual materials used in the equipment stainless steel with the same finish, polymers, glass, etc.
Samples of the equipment surface.
Yeah. Then they carefully apply a known amount of the API solution onto the coupon surface, that's the spike. They let the solvent evaporate, leaving a known residue amount.
Mimicking dried-on product.
Exactly. Then, after a set time, they sample that spiked surface using the exact same swabbing or rinsing procedure planned for the actual validation.
And analyze the sample.
Yes. The lab measures how much API was recovered. That amount is expressed as a percentage of the known amount originally applied.
What's a good recovery rate?
The plan says 75% recovery is generally acceptable. Maybe down to 50% for tricky surfaces like some rubbers, if justified. It proves the sampling method is reasonably efficient.
So the lab result reflects, say, at least 75% of what was actually there. Good to know. Now,